back and I'll give you guys an introduction to the lab activity for today. I'm gonna discuss a bit of history of how we used phenotypes to describe bacteria. And although these aren't necessarily used as much to identify specific bacteria anymore, a lot of these same methods are used to understand the basic science of how bacteria grow and divide and colonize plants. What we're moving towards a lot for bacterial taxonomy is the one phenotype we use is, of course, plant disease outcome. If you're familiar with the disease, then you probably know what you're looking at. But when you're unfamiliar, where we're starting to go more and more is into the DNA level and identify bacteria based on their genome sequences. Bacterial taxonomy and phylogeny is challenging. It's still a surprisingly controversial topic. And if you want to get some people, some professors really worked up at a conference, sometimes bringing up bacterial taxonomy is the way to do it because people have strong opinions about how we should be classifying bacteria. And some of it is that there isn't a simple answer of how we should classify bacteria. So species, any sort of species that we define, like even if we're looking at tigers, that is a little bit that humans have decided where to draw the border for the species of tigers, because tigers and lions can crossbreed and you can get a liger. So, some of it is that species are, not as neat as we'd like to classify in our textbooks, but nonetheless, being able to classify things has value in terms of being able to know what we're looking at and communicate about what we're looking at. And so there's a lot of attempts to describe bacterial species. And bacteria are difficult because the classical species concept had to do with whether individuals can interbreed. And bacteria generally are transmitting most of their genome vertically. And then they can also interbreed, do horizontal gene transfer with very distantly related bacteria. So where those classical species concepts and species definitions are described, bacteria don't fit very well. But nonetheless, bacteria definitely do have species, and the debate is mostly like a little bit semantics, and it really has to do with where you draw the line at like 98 or 98.1% similar. So there are conventions that, as we get better and better technologies, we're, drawing the line with more confidence. And what makes bacteria also difficult to classify is that they don't have many morphological features. So, before people knew as much about cats and tigers, it still was like pretty clear that they were different species. But with bacteria, it's taken a lot of descriptions and a lot of studying of bacteria to know where to draw borders, because so many bacteria will look like little pink rods when you stain them with the gram stain. There'll be a lot of gram-negative rods. And so trying to define, that bacterial species is a little bit harder. Okay, but nonetheless, we've focused a lot on phenotypic traits and genotypic traits. So we highlighted the gram stain before. This is it's still a very useful approach, although we don't always there isn't a lot of using this approach to identify bacteria, but this trait of their cell envelope corresponds to their evolutionary origins, and so it's been very useful to keep these names of gram positive and gram negative because it corresponds to that cell envelope biology and also to the evolutionary origins. Morphology was also used to classify bacteria and identify them. And there's potentially a lot of different morphologies and attachment arrangement that you can see. But plant pathogens are kind of boring. For the most part, there are just so many gram-negative rods. And so for like medical micro, you can see Neisseria that causes meningitis and gonorrhea. Like there's a lot of really cool, like, clustery ways that a lot of the human pathogens grow. But for the plant pathogens, it was a lot of gram-negative rods. And so this wasn't super useful for classifying most bacteria. But there are there's other structures where there are the streptomyces that are filamentous, and then the spirilla shaped phytoplasmas. So there's at least a little variation, but not enough to be a super useful way to classify. For a while, motility traits were a way that people would try to classify bacteria. But setting up these assays takes a lot of time and it's not very practical. But it is a cool way to study bacterial behavior. So this is it's soft agar. So it's a semi-viscous media, and you stab bacteria into here and you can see this one doesn't swim. So it doesn't move very far. But if bacteria are able to do swimming motility, they end up moving throughout the whole media so that they can consume all the nutrients and they grow cloudy and turbid. And so it's a way to look at bacterial behavior, but not that useful for classification. Media traits can be useful still, especially if you know what you're looking for. There's a lot of different words that have been systematized as far as how to describe bacterial colonies. So my favorite bacteria is Ralstonia, and it forms irregular colonies, and maybe we don't really describe it this way, but probably more undulate edges. And I would say, it makes pretty flat, goopy colonies for Ralstonia, maybe a little bit raised. But depending on how the bacteria, what traits the bacteria express, their colonies on plates will look a little bit different. So when they're really shiny, that's usually because they're making polysaccharides that are, a little bit goopy. And so depending on whether they're making a lot of polysaccharide or not, they might look smooth and shiny or dry. Dry would be without polysaccharides. And so if you're trying to grow bacteria out of plants, there's different approaches you could do. So media is also known as agar. So in your petri dish, you'll put agar and different nutrients. And depending on what nutrients and other chemicals you put into the media, you're making an experimental choice about what you're trying to grow. There is really no such thing as a truly non-selective media, but there are rich medias that allow some bacteria to grow without too much stress. But there's no universal media that can grow every bacteria. If you have a suspicion of what you're growing, people have defined specific medias that can help you pull the right needle out of the haystack. And these can have different antibiotics in them. Sometimes if you're focusing on bacteria, you'll put fungicides so that you don't get fungi growing out. And so that can help you narrow down what you're looking for. And then there's also compounds that you can add to media to differentiate. So this doesn't change who grows, but it changes how the bacteria will look on plates. So sometimes different dyes, including crystal violet, which is used in the gram stain. Crystal violet's used. There's a red pigmented compound that we use for Ralstonia. And this just changes how the colonies will look when they grow on that media. So it gives you a little bit easier of a time distinguishing whether you think it's the right bacteria. There's also ways that you can classify certain bacteria by whether they grow in an oxygen environment and whether they are able to grow without the presence of oxygen. But that's a pretty old school technique, at least for diagnostics. And then finally, if you if you know what you're looking for, there's certain characteristics that are present in a lot of clades. So there's a lot of Pseudomonas that secrete compounds into the agar. Some of these will be fluorescent, and we call those often Pseudomonas fluorescens. But that's one of the, like, Pseudomonas is a complex genus that is, there's a lot of strains that produce fluorescent compounds that are not Pseudomonas as well. But if you see that fluorescent compound, you can create a hypothesis that it's Pseudomonas. A really convenient one, xanthomonas are almost always yellow to orange colored. So those are a little bit easier to pick out. Like if you see disease that looks like xanthomonas, you plate it out and you see a lot of yellow colonies, it's probably the xanthomonas disease. But most, even when we go to these classic phenotypic descriptions, the descriptions are not super useful for being able to quickly identify bacteria. We have a lot of similarities in terms of being rod-shaped, gram-negative, whether they have a flagella, and that's hard to look at. Sometimes we can get certain distinguishing characteristics, but it's not very good for being that accurate. People developed some more high throughput methods for looking at phenotypes. Some of these are in a microplate format where there are, say, 96 different carbon sources in each well. And depending on which metabolite the bacteria grows on, you'll get production of this purple compound. And you're able to really, like, if you have a huge database, you can match up the correct thing. That's not super useful, IMO. Another way that some people use to identify bacteria is based off of their lipid membrane composition. Every bacteria has slightly different lipid composition, and so this can be a reproducible way to identify bacteria. Antibodies have been used to identify certain surface features. So like whether they have some of the antibodies are reactive against the pili or the EPS, extracellular polysaccharides. And so those can be used to identify bacteria. But I'd say the two most useful for identifying plant diseases is to have an idea of what you're looking at. And so some of this depends on where you are in the world, which bacteria diseases are common there. And then exactly how they look. So these are different Pseudomonas syringae strains on the same tomato plant. And you can see that there is a lot of natural variation in how the disease manifests. Sometimes you get these blackening, these brown pigments in the middle of the lesions. And sometimes you just have really subtle chlorosis. So that can be useful for distinguishing strong versus weak pathogens. But I'd say if you want to identify your plant disease, the best thing is to get some resources. There are a lot of compendiums through the American Phytopathological Society that focus on specific diseases, as well as look for, if you're concerned about your home garden, one of the best bets is to search for extension resources at the local universities. So UC Davis has a lot of extension, and see what kind of plant pathogens are common in your area. Okay, so if you're not sure exactly what you're looking at, DNA-based methods are one of the fastest and efficient ways to identify the bacteria. And someone said earlier, differences between, when we were talking about differences between bacteria and eukaryotes, they highlighted the difference in ribosome composition. So bacteria have a different ribosome structure than eukaryotes. And one of these components is what we call the 16S ribosomal RNA. So this is, the S stands for Svedberg's, which is where it goes in a centrifuge when you put it in a density gel. So it's a very old school way, but you don't have to know the details of that. You just have to know that it's the 16S RNA gene. And this has been used as a marker for bacterial taxonomy since the late 70s and 80s. It is a housekeeping gene, so it's present in I think all bacteria. I don't think I've actually heard of any cases without a 16S gene, but there's probably a weirdo. All bacteria have the 16S gene. And so it's a gene that encodes a basic function of bacteria, encodes part of the ribosome, so part of the translational machinery. That means it mutates very slowly. There's a lot of selective pressure to keep it behaving similarly for the bacterial cell. But nonetheless, it does accumulate mutations through evolutionary time. And so if we compare the DNA sequence of that gene between different bacteria, we can create something like this phylogenetic tree over here, and see that some strains are more closely related to each other, and then are very different, start to get very different if we start going out these branches, start to have a lot more diversity. And so we can look like the activity that the lab today is going to focus on using 16S gene sequences to identify 6 unknown bacterial plant pathogens. And you'll be using the NCBI BLAST database to do that. In addition to just studying a single gene, there's approaches to study 3 to 5, maybe up to 10 genes with this MLST approach, multi-locus sequence typing or analysis approach. It's basically the same. You sequence multiple genes and then you make a more confident phylogenetic tree. And this one's not very important, but if you end up becoming a microbial ecologist, you might see this approach a lot in the 90s and early 2000s. It's not too expensive and hard to do, and it's not worth it. But it is an approach that still looks at those genes, like the 16S, but instead of sequencing it, they use enzymes to digest the pattern onto gels and then look at patterns. It's a way to look at bacterial diversity. And currently we're in a pivot point where sometimes we will use single DNA approaches, single gene approaches, but more and more we're going to whole genome shotgun sequencing approaches. This has already happened a lot in clinical microbiology for human diseases, and more and more plant pathogen approaches are going that way, especially where there's some new emerging outbreak. That's a new disease we haven't encountered before. And we need to understand its ecology to define management approaches to contain that plant disease. There's, I didn't quite say that. So there's multiple approaches to sequence bacterial genomes. You'll hear the different technologies, Illumina, PacBio, and Nanopore. Those are approaches to sequence all of the DNA in a sample. And then once you have those DNA sequences, you can compare them to each other or compare them to databases. And the approach that's used is this, that's common is the average nucleotide identity approach, which is similar to BLAST that you'll be doing in your lab, except it's comparing usually at least 1,000 genes between 2 different bacterial genomes to identify how similar they are to each other. And it's only comparing the genes that are shared. So this is more for the grad students or the bacteria aficionados. So if we compare multiple bacteria strains, they have variation in their genome content, but we can define what we call the core genes. So these are the genes that are shared between multiple strains. And those are shown in blue. And so as we add more bacteria into our comparisons, you can see that the number of core genes goes down the more comparisons that you start making. But at some point it plateaus and you get a certain number of genes that are usually present in all strains. And then you start to see this almost, it will plateau eventually as well, but you get this, this increase in what we call the pangenome, which is any gene that's present in at least one strain as you start comparing these. And when we're doing whole genome comparisons, we're usually focusing more on this core genome. So you'll see that a lot of these bacteria have a very high nucleotide identity on this core genome, but they might still have, like if we're comparing these two, these ones, there might be, a difference in plus or minus, like presence, absence of hundreds of genes between two bacterial genomes. And so I just want to highlight that for the grad students, especially because it took me a long time of thinking about average nucleotide identity before I understood that it was focusing only on the similarities and was ignoring how much strain to strain variation there can be between two genomes. So it's useful for taxonomy, but not necessarily for predicting function, and not necessarily useful for predicting disease outcome for your plant pathogens. Okay, so for today's lab, you'll first watch a few videos that describe how you might isolate bacteria from an infected plant, and then it'll move into a portion that walks you through how to use the BLAST database to identify a 16S sequence. So I'm going to give you multiple unknown DNA sequences from our unknown samples that came into the disease clinic. And these DNA sequences have come from the 16S gene. So this is what the 16S ribosomal RNA looks like. You can see it's got all this really great base pairing and secondary structure. And then there's these variable yellow spots. So those are the variable regions that are useful for classifying the genetic diversity of bacteria. And in this case, I kind of just pulled sequences randomly out of NCBI, but we can pretend that it's this DNA amplicon in this part of the gene that spans some variable regions. Okay, and so as a reminder, the 16S gene is only present in bacteria. There is a ribosome in all organisms, but archaea and eukarya have a different RNA, and it's an 18S that has a lot of similarities, but the archaea and eukarya RNA will not amplify in a PCR reaction. Okay, so what the lab's going to do is walk you through how you would streak bacteria onto plates. So one thing we're going to ask you to do is, in some sort of way, paper and pen and take a photograph of it, use Microsoft Paint. We want you to draw a petri dish and use your mouse or your pen to, describe how you're going to streak out the bacteria in order to get these single colonies. And as you streak the bacteria, you would change your inoculation loop or your toothpick, either flame it or get something new that's sterile so that you can slowly dilute out the bacteria in order to get a single colony. And this colony would have derived from a single cell that landed on that agar and then grew for 24 to 48 hours up. So it's a little homogeneous population of bacteria. And there'll be a video that'll help walk you through that, so you don't have to remember exactly what I just said. And so once we have our single bacterial colony, what we're going to do is extract the genome from it and do a PCR reaction that amplifies its 16S gene. And so a PCR reaction will helps you amplify specific DNA fragments. In your PCR mix, you put your genomic DNA, your DNA sample. You have primers, which are short pieces of DNA shown here in red. They have, depending on what you're trying to amplify, you choose specific primers that have a DNA sequence that matches your target DNA. The reaction also has to have nucleotides so that we can add in nucleotides during the reaction. There needs to be an enzyme called a polymerase that amplifies DNA. And then you also would add buffer, which has the right pH for the reaction and the right salts that help everything move forward. And so you would mix all of these things together and put them in a little PCR tube and put them in this thermal cycler. The thermal cycler will then go through multiple temperature cycles up to near boiling, which allows the DNA to melt to denature. So that allows all of these base pair hydrogen bonds to disassociate from each other, and so that you separate the two strands of your DNA. So then you have single-stranded DNA, and you cool the temperature down to 55. And at this cooler temperature, then it can allow base pairing to happen again. So ideally what happens here is you get base pairing between your specific DNA primers into their correct spot in your target DNA. And they bind at the annealing step, and then you heat the reaction back up to 72. This is the optimal temperature for those Taq polymerases for them to start chugging along on the DNA, incorporating the DNA nucleotides and synthesizing a new strand of DNA. So during this extension phase, you get the DNA synthesis happening and the extension of that DNA. So at the end of the first round, you now have 2 pieces of DNA where you originally had 1. And so this will then go through 25 to 35 cycles of this temperature from 95 to 55 to 72, and you get an exponential production of your target DNA. And I think it's really important to know that the primers that you put into the reaction are what determines what DNA gets amplified. So if we were targeting the ribosomal DNA, we would be, I've drawn them on with like this red marker. Our primers would, we'd probably have a forward primer that goes in this direction, and then a reverse primer that's on the other end that points, they need to point towards each other from 5' to 3'. And so by putting those 2 primers into our PCR reaction, we should be able to amplify this 16S gene. There are multiple approaches that people have used for 16S DNA sequencing. So if you Google, you might find primers that bind to this one. But you get to determine what primers you put into the reaction. So after we do our PCR reaction, what you need to do is determine whether your PCR worked. And you can do this by running it on a DNA gel through DNA electrophoresis. And with this, you would pour, if you're in lab, we would pour gels composed of agarose, which is very similar to the agar that is in Petri dishes to grow bacteria, but it's a little bit more pure and a little bit more expensive. And so you would pour this and let it polymerize, and you would have a comb that would have gone into the top of the gel. And this just serves to make little divots in the gel so that we can load our DNA sample. So you would then take your PCR reaction and load it in with a dye that makes it heavy and fall down to the bottom of the well. And it would, your DNA would sit there. You'd put your samples in a few of these wells. And in one well, you would make sure to add a DNA ladder. And that allows us to estimate the size of our DNA. So now that you've loaded your gel, you'd put the lid on it. You'd read the danger warnings because we're about to run a lot of electricity through our gel. And so they're designed to not, usually not allow you to stick your hand in the buffer. You would never want to do that. You'd probably get a good shock. And we'd close it up, and then this gel system would run an electrical current through this running buffer. And it would run from negative at the top to positive at the bottom. And the DNA backbone has those phosphate negative charges, And so by having a positive charge at the bottom of this gel, your DNA would migrate through the gel towards that positive and separate based on size. Because the DNA is going through this gel-like matrix, as it goes towards the positive, the smaller pieces of DNA are able to move faster through the agarose. The larger pieces of DNA are longer, and they're kind of like a Labrador with a big stick getting through a door. They get stopped at a lot of the pores. And they get held up, and it takes them longer to migrate through the complex gel matrix of the agarose. And so you get this separation. So after running it, and usually there's these dyes in it, so you can estimate how far everything's gone. You're not actually seeing DNA bands, you're looking at loading dyes that help you make sure all of this DNA doesn't run off into the buffer at the end. So you'd run it for 30 minutes to an hour, and then we'd go put it on a gel imager and see what our success was. In our DNA gel, we'd have to have some sort of additional dye that allows us to see the DNA. This could be ethidium bromide or any other dye that is fluorescent and fluoresces when it's inside of double-stranded DNA. So ethidium bromide will, the molecule itself intercalates between the DNA bases and fluoresces when it's inside DNA. So you get these fluorescent DNA bands wherever there's DNA in the gel. With this gel, we would see the DNA ladder. And so this is something we buy from companies that has DNA of the correct size. And then this would be our PCR reactions. So we'd see this one and be like, okay, that PCR reaction is 1,000 base pairs. In this sample, we have an 800 base pair and a 200 base pair gel or band. So you always, when you're looking at a gel, you want to see, did you get a band? Did you expect to get a band? And if you got a band, what size is it? And is that the size you expected? Those are the questions you always want to ask yourself when you're looking at a DNA gel. And this part we would not get, you don't get to experience, but in the real lab, your PCR might not work. In our online lab, your PCR worked. So if you get really messy gels like this, you're not alone. There's a lot of things that you could be doing better. So I recommend Googling. There's a lot of advice on how to, not get these smears, not get, oh, maybe we have a non-specific band and we need to design better primers. Maybe we should clean our gel comb better because there's chunks of agar that's making our ladder run weird. These are all things that can happen. So then once we know that our DNA amplification was successful, what we would have done is submit that DNA for Sanger sequencing. This is a different sequencing technology than the genome sequencing technologies. So genome sequencing we call next generation, and there's 3 companies right now that are leading that. Those are Illumina, PacBio, and Nanopore. Those might change another 5 to 10 years. They might come up with even better approaches for sequencing DNA. But all of those approaches are really focused at genome level. This approach of Sanger sequencing, although it's a very old school technology, is still, I don't know when it's gonna become outdated, because it's one of the faster approaches to find out a very small amount of DNA. And so it's actually, it's still very cost competitive and a pretty fast turnaround time. So this is what we would be using to sequence a specific DNA. And with this technology, you're able to sequence about 800 base pairs to 1,000 base pairs with high accuracy. And so that's where it's useful for doing single gene analysis, but also if you're doing biotechnology and genetic manipulations, this approach is very useful for checking what happened in your reaction and making sure it isn't just a hairy mess. Because there's a lot of ways that cloning can go wrong. And so the way this technology works is very similar to PCR. You similarly get amplification of DNA, but there's one difference in the reaction in terms of what gets added into the mixture. And I'm gonna go through the details of this. Realistically, you will never put together this reaction, even if you do a lot of Sanger sequencing. Usually you just give someone else your DNA and the primer that you want to use, and then they do this whole reaction. But it's worth understanding how it works. So with this approach, in addition to having normal DNTPs that are the base pairs that get added in the amplification, there's a handful of DNTPs that have a fluorescent dye attached to them, and they're missing one of the hydroxys. So they're, instead of being deoxy nucleotides, they're dideoxy nucleotides, and they're missing both of the hydroxys, hydroxyls. And that means that they will terminate the chain. So these fluorescent dyes, once those fluorescent, like that fluorescent G gets added, at that point, the DNA polymerase has nowhere to add the next base pair, and it terminates the reaction of that piece of DNA. And so depending on where this the fluorescent dye gets added, once that happens, it ends the reaction, and you get it ends the extension. And so after 35 cycles, you end up with a population of DNAs where each one has a different termination base pair at a specific point. And then there's a DNA sequencer that is a very expensive gel electrophoresis machine. It runs all of these fluorescent DNA fragments through a very long, it's like meters long, all wound up inside the sequencer. But it's the exact same thing as gel electrophoresis. And it runs off the shortest one first. So that one comes out, then there's a laser that detects, that fluoresces it. And it detects what color fluoresces. And so if it's the color of the T, then it records that as a T. And so as the DNA comes off, you get these spectra bands that increase. And so from that, you actually, when you get your result back from the sequencing company, they usually give you a file that you can visualize like this and see that there's a really nice G band, a really nice A band, and you can be confident in the DNA sequence. But also sometimes it's a little bit messy. So it's good to look at that and make sure that you don't have a garbage reaction. So once you have that spectra or the DNA sequence chromatogram, you're able to extract the DNA sequence from that file and ignore all the bands. And you end up extracting, your sequence that's GC, GCC. And you can put it in a bioinformatics format that we call FASTA, F-A-S-T-A. It's one of the simplest bioinformatic formats. It just has this little caret, any sort of sample name, and then a line break to the DNA. And it's worth knowing about that format because if you do anything with DNA sequences, you'll probably put your DNA into that format at some point. And you're able to put multiple of these after each other. And then the bioinformatic programs know that it's a new sample when they see this sideways caret, the greater than sign. Okay, so then you have your DNA sequence in FASTA format. And we can use the NCBI database to identify what that DNA, what it is. In this case, we already know that we put primers into the reaction that amplify the 16S gene. And so we're going to be searching our DNA against the 16S DNA database. So we paste in our sample into this box up here. It is our query sequence. So query is the thing that we're searching against other things. In yellow here, you'll have to change this dropdown so that it matches the 16S database. There are other databases that you can use when you have different scientific goals. But right now we want to find out our match in this database. You'll hit submit somewhere down here, and then you'll go grab coffee and come back, but a really fast coffee, because it'll be like a minute and a half. And when the results page comes back, you'll see something like this. This has been like, this is what it looks like for a few years now. At some point NCBI will probably change the interface, and you'll have to figure out where to look for all of your results. And we'll have to update our tutorials. But for right now, you'll get information back like this. I encourage you to explore around in this and just, click different links and explore it more deeply. Most of what I've learned about bioinformatics is just by doing the approach and then clicking to see what the bioinformatics scientist thought was useful to display to me. But for the purposes of this lab, we'll be looking at what the top hit is. So in this table view, it gives us a description of the organism that our blast hits correspond to. And what I like to focus on is the percent identity. So that's a pretty low percent identity. It's 93%. For most of you should be getting much higher than that, but I decided to do the weird one as the example in the homework. It's worth looking at the percent identity, and also right here, the query cover. That's short for coverage. That shows just how much, did it match up the whole gene? So in this case, we got 100% coverage to it. But if you see a very low percent there, that indicates that your results, don't trust your result much. Don't trust something that's 35%. So it's worth always checking the coverage percentage and just making sure that you trust what the database is giving you back. And what we're asking for in the worksheet is for you to give us the genus and species of the hit and the percent identity. We don't ask you to do too much more. But one thing I would like you to do, encourage you to do, is click on this. I was highlighting that you need to do percent identity. I want you to click also on this alignments tab, and go ahead and look at the nucleotide alignment. This shows you where that 93% identity comes from. So query, this is the DNA that we put into the database, and then this is the thing that came back out. The match in the database. So query is what we searched with. And when you look at these pairwise DNA alignments, it shows you the base pair match, these are not base pair matches. These are identical matches. So you'll see a little line where the base pairs are similar. And then when there is a lack of alignment and you don't have the same sequence, you get a gap in this middle region. And so here we can see that C and T are different. There's not a line there. And additionally, the thing in the database has one less base pair. So where there's a little gap, you'll have a little dash. And our query had one extra base pair there. So I just want you to look at this, because it's always good practice to look at these, especially if you've looked at plenty of alignments, you don't have to explore this too much. But go ahead and look and see for something with a low identity, just get a sense for oh yeah, I agree that there's a lot of mismatches there. And if there's a really high identity, you'll see that the sequence overall really aligns and matches up well. And it's not the focus of the lab here, but there might be times where you do amino acid sequence alignments further in your careers or your education. And what's really interesting to look at in amino acid alignments are places where sometimes if you compare two proteins, you have a lot of sequence being similar, and you're like, okay, it's all really well aligned. And then you get really variable regions. So that's usually means that there's some very interesting biology in that variation region. So that's a reason to get used to and excited by looking at alignments. Yeah, and so then in lab, focused on these values though for the answers that we're going to grade. The genus and species name and the percent identity. And then, yeah, if you get something that's a very low identity, then you probably searched wrong. So come to office hours or email us if you get really off. But all of your things should be high identity. And then the final step of the worksheet is to make it, interesting for a plant pathology lab. So now that you have a table of your potential bacteria hits, there's going to be a table that has different names of plant diseases. And we've given links to a couple reputable databases for plant disease pictures and diagnostics. And I want you to match your bacterial pathogen to the correct disease, and then pull out an image from one of those image databases that shows symptoms of the disease, and describe at least one symptom in written words that describes that disease. Yeah, and then that's going to be the lab. So we'll probably stop the recording there, and if people have questions.